So a couple of you asked to do a DNA gel example and asked why, when we're running this large DNA gel, why do the larger fragments, for example, say that this is our ladder? Why do the larger fragments, say that you load up here, why do the larger fragments get stuck here while the smaller ones can make it down here? The medium ones get about this far. So as the current drags your DNA down here and it's attracted to that charge, the smaller pieces can wiggle through the gel sugars more quickly than the big pieces can. So if we go down to the molecular level, what these gels are made of is these large sugar chains like this. And so, when you're a small DNA molecule, say you're only 100 base pairs long, you can just slip right through here and you're gonna move right along towards the current. However, if you're a large DNA molecule, tough here, you're gonna kind of get stuck right here, you're gonna have to like fold in, it's gonna take a lot longer to get even just over the same amount of time and same charge, it's gonna take a lot longer to move an equal distance. So in this case, the larger DNA molecule, it just can't fit past these little sugar molecule chains that the gel is made of, and it just gets stuck here, while the smaller molecules can speed right through in between the sugar molecules down there.